Good afternoon. My name is Regina Hendricks. I'm from the Medical Lab Technician Department. I will be presenting from slide 30 to 39. The KEL antigen system is a group of antigen on the human red blood cell surface which are important determinants of blood type and targets for autoimmune and autoimmune diseases which destroy red blood cells. KEL can be known as K or Kp. The KEL antigens are peptides found within the KEL protein. KEL antibodies produced as a result of immune stimulation. IgG reacts well at AHG. Anti-K occurs much less frequently due to high frequency of antigen. 1 in 500 people are K negative. Donor units must be antigen negative cross-match. Anti-K is the most common because the K antigen is extremely immunogene. However, K, Kp, and JSB antibodies are rare, but a lot of humans have those antigens but won't develop an antibody. Myeloid cytosine is an X-link, a normal of the KEL blood group system in which KEL antigens are poorly detected by laboratory tests. The myeloid gene encodes the XK protein, a protein which is structural characteristics of the membrane transport protein of an unknown function. Questions that you may ask yourself: what antigen is associated with myeloid cytosine? The myeloid cytosine is characterized by an absence of the KX antigen on red blood cells. This is associated with weak expressions of KEL antigens. KEL antigens are connectively bound to red blood cells. Transmembrane XK protein. When XK is absent on red blood cells membrane, myeloid cytosine is developed. KID antigen enhanced by enzymes. Not very accessible on the red blood cells membrane. The membrane of the red blood cells plays many roles regulating their surface: deformability, flexibility to other cells, and immune recognition. KID antigen, anti-JKA and anti-JKB, has IgG. It usually appears with other antibodies when detected, while anti-JBB is found in some individuals which are JKA negative and B negative. Questions that you may ask yourself: what is JKA antigen? The KID antigen system is also known as JKA antigens, or proteins found in KID blood groups, which act as an antigen. They have the ability to produce antibodies under certain circumstances. Another question you may ask yourself: how do I get anti-JKA antibody? Antibodies to red cells antigens are considered naturally occurring when there is no obvious source stimulators, such as blood transfusion. These antibodies are produced as an immune response to some unknown environmental antigens, such as polygrains and other parts of bacterial membranes. And to wind up my presentation, Duffy blood group. Duffy blood group system antibodies. Anti-FYA was first described in the serum of the multi-transfused hemophilia patients. Identification of FYB antigen followed in 1951. Antigens are destroyed by enzymes. Duffy blood group. Duffy antigen, which is also known as FY glycoprotein, is a protein that in humans is encoded by the ACKR1 gene. Duffy antibodies are predominantly of IgG subclass, whereas the IgM form is rare. They do not react with enzymes treated red blood cells. This is the end of my presentation. I hope you guys enjoyed. Thanks for watching.